We did see data on CC220, and many of you guys, I think, participated in that trial. That's the newest of the IMiDs, or the CELMoDs, which is the new hip name for them. And it's basically a more potent cereblon binder than thalidomide, lenalidomide, or pomalidomide. And what we showed at this meeting was a response rate of about 30%, with actually what I will say was a pretty modest adverse event profile. So it's an oral approach, weekly dexamethasone, not a lot of GI tox, not a lot of confusion or fatigue, as you can sometimes see with the IMiDs, particularly in refractory patients. And heme toxicity was really the biggest issue that we saw, and well above now the dose that we expected to be the maximum dose that we would get to. And now combining with DARA, combining with bortezomib, combining with carfilzomib, really to see how to best think about bringing this agent forward. I don't know if you guys have had any experience.
No, so we are latching on to this study with some immune correlatives, as a sub-study. I think what was interesting is the response with each dose. So, there were different dose levels, and you were seeing responses even with the lower doses, I think at, as low as 0.45. So it would be interesting to see what's really happening, which mechanism of action is iberdomide relying on at each given dose, whether it's more immune-mediated versus direct cell kill. I think it would be important to understand that so that we can partner it with the right therapies moving forward with so many interesting targets that we're looking at. So I was very impressed with the data that you presented, Sagar.
I was very impressed in the lack of toxicity. That was amazing, actually. In fact, most of the people who are on long-term lenalidomide all get some degree of GI toxicity. And I've always assumed that that's from some immune-related effect. And I thought the CELMoDs actually were going to have more of kind of an immune suppressive or immune stimulatory effects and would potentially have more diarrhea. But this showed just the opposite, which is great.
So we've covered a lot of ground, a lot of different areas and a lot of different exciting data from ASCO. Do you each want to give me sort of your take-home message, what you may do differently, what you may not do differently, what you learned, short version? Why don't we start with Saad?
So the key message from the large randomized phase 3 trials, I will have a conversation with my high-risk smoldering myeloma patients now about treating them based on the data that were presented. I left augmented with the feeling that transplant is still the standard of care for frontline patients, as is the use of a PI-IMiD-based triplet regimen. And I'm hoping that the sub-Q data actually leads to an approval, and then we'll have to figure out how each of the healthcare system deals with the IV versus sub-Q formulation administration. But I think it's good for the patients. So those are my key takeaways from a clinical practice standpoint.
Great. Karina?
I think, we covered so much even at the orals and all the posters, but I guess I'll stick to more of the relapsed refractory since you did the smoldering and newly diagnosed. We have so many options, and I think we have more questions now than answers, but I think that's great for myeloma. The fact that we have more and more, and that we are sort of on this cutting edge immune therapy versus targeted, therapies. And myeloma is so different than everything else. And that's my biggest message, that I think, venetoclax, all these other drugs that work so great, and we have to be different with our myeloma patients. They do have different side effect profiles. Same thing, CAR Ts are not a cure for our myeloma patients, but our myeloma patients haven't had a cure. So when we look at this data, as myeloma doctors, we understand that. But I think as community doctors, sometimes we're saying, oh, this isn't really working on these patients. In my opinion, everything that was presented was actually really, really amazing. These response rates of single agents for BiTEs, for CAR Ts, for even venetoclax, even though it's an FDA hold, are actually really, really promising.
Yeah, I would agree that the oral session was a bit like taking the MCATs.
Yes.
There's a lot of interesting stuff there. Marita?
I think Saad and Karina sort of covered it most just in terms of promise. I think for me as a transplanter, I'm still very happy with the Forte results that, okay, we can go home, we still have a job, that transplant, deep remissions, long remissions, higher MRD. And I think I share the excitement with the multiple immunotherapy approaches, but I'm waiting to see more BiTEs than AMG 420. I don't think that's going to be the answer.
Mm-hmm. All right, Tom.
So I think the meeting has been amazing, actually, with all the data that we've seen. However, I'm going to take the future approach, and sorry, Amrita, you might not like what I'm going to say, and that is I do think that the antibody therapy is going to be combined with an IMiD and a proteasome inhibitor. So we're going to do quads as induction therapy for myeloma. I think we're going to get deeper remissions from quad-based therapy, and I don't think we're going to do 4 cycles. We're going to do 6 to 8 cycles, and at that point in time, we are going to evaluate MRD. And MRD, in my mind, has to be 10 to the minus 6. And then we're going to make decisions based on MRD on whether we're going to do transplant or not. And there's going to be some people that don't need a transplant.
So Tom, you're describing one of our investigator-initiated protocols right now.
Which is perfect. I think that's where we're going to be. But then we have so much excitement with these new drugs, like the bispecific T-cell engagers and CAR T-cells. And they are, in fact, in people that are really refractory, inducing deep remissions in MRD negativity. That is unbelievable. Anything that has such a potent effect in the relapse and refractory has to be brought sooner on in the therapeutic realm, either as part of induction, consolidation, or maintenance, or as first relapse, early relapse. And I think as we bring these drugs further frontline, that, again, transplant's going to start going away because we're going to be able to do it without doing a transplant. We're going to be able to induce these really deep remissions.
We just have to remind Tom that in CAR T cells, people achieve MRD negativity and they still relapse.
Well, they do a transplant.
Yeah, it's
Not
not
a cell
about
mod with
one
it.
time
Fine.
point. It's
I
about
do too.
sustained
Correct.
MRD
Correct.
negativity.
We agree.
You
It's
know?
about sustained MRD
There we go.
negativity.
All right, good, good. All right, well, thank you all very much. And I think this has been really a great discussion. On behalf of our panel, we thank you for joining us on this and hope you found this OncLive peer exchange discussion to be useful and informative. And thank you for your attention.